Hi, I'm Ron Benitez. I'm with MilliporeSigma, the life science business of Merck KGaA, Darmstadt, Germany. I am an immunoassay specialist here in San Diego, California, USA.
Hi, my name is Jayana Barrett. I'm with MilliporeSigma, the life science business of Merck KGaA, Darmstadt, Germany. I'm an account manager and specialist in San Diego, California, the United States.
And I'm Alex Coe. I'm with MilliporeSigma, the life science business of Merck KGaA, Darmstadt, Germany. I'm a field application scientist in San Diego, California, the United States.
Today, we're discussing how to build a standard curve using serial dilutions. So what is a standard curve and why is it important? Standards is a series of known samples with known concentrations, with which measurements are used to build a curve to interpolate unknown sample concentrations based on their measurements. Standard curves are important because they allow for accurate measurement of unknown samples. Curves can be used for measurement of many different types of chemical and biological molecules, including proteins, DNA, and inorganic purities. What are serial dilutions? Serial dilutions are an accurate method of generating standard curve samples with a wide range of concentrations.
So we're going to go ahead and set up the standard curve for you. So what we're going to need is we're going to need a chemical that we're going to measure, and we are going to need a pipette. We're going to need pipette tips to transfer the diluted standard, and then we're going to need tubes, these tubes obviously, to hold the diluted standard. And then we're also going to need a method to mix the tubes, which you can do it by hand, which we're going to do today, or you can use a vortexer.
So now we're going to set up a serial dilution of this chemical standard here. We're going to do 4 subsequent serial dilutions. Each serial dilution is going to be 5-fold. So in this 5-point curve, the lowest standard point in the curve will be a 1 to 625 dilution of this original stock here. To perform our serial dilution, we will first load our P1000 pipette with a fresh tip and transfer 1 ml of the original standard solution into a tube to which we had already added 4 ml of dilution buffer. This will of course be a 1 to 5 dilution as indicated by the label. We will then close the tube and invert it to mix thoroughly. We will then continue our dilution series by using a fresh tip to transfer 1 ml of the 1 to 5 standard solution that we had just prepared into the third tube that contains 4 ml of dilution buffer as well. This is our 1 to 25 dilution, which we will also mix thoroughly. At this point, we will perform 2 additional serial dilutions, using a fresh tip for each transfer step and mixing thoroughly afterwards to prepare the 125-fold and 625-fold standard dilutions, thus completing a 5-point standard curve, which, along with our blank solution, can be used to accurately measure concentrations of our molecule of interest in unknown samples.